Understanding how cells evolve over time is one of the most profound challenges in modern biology. From embryonic development to tissue regeneration and disease progression, the journey of a cell from one state to another is not a straight line, but a complex, probabilistic path shaped by gene expression, population dynamics, and the microenvironment. But here's the catch: we can't watch cells change in real time. Single-cell RNA sequencing gives us snapshots, static views of thousands of cells at different time points, but no continuous record of their individual trajectories. So how do we reconstruct the invisible journey? The answer lies in computational modeling. But most existing methods fall short. They treat cells as isolated points in high-dimensional gene space, assume constant population sizes, and ignore the fact that a cell's fate is influenced by its neighbors. They're like trying to predict the weather using only temperature readings from a single station, missing the full picture. Enter MIOFlow 2.0, a unified framework that integrates manifold learning, optimal transport, and neural differential equations to learn biologically plausible cellular dynamics from time-resolved single-cell and spatial transcriptomics data. At its core, MIOFlow 2.0 addresses three fundamental biological realities that previous models have ignored. First, stochasticity and branching. Differentiation isn't deterministic. Two cells starting in the same state can follow different paths due to noisy gene expression and probabilistic fate decisions. MIOFlow 2.0 captures this using neural stochastic differential equations, a mathematical framework that models both a deterministic drift, the developmental program, and a diffusive noise term, transcriptional variability. This allows the model to generate a distribution of possible trajectories from a single initial state, naturally representing branching events. Second, non-conservative population dynamics. In real tissues, cells don't just move, they divide and die. Populations expand and contract. Standard optimal transport assumes mass conservation, like water flowing through a pipe with no leaks. But biological systems aren't closed. MIOFlow 2.0 introduces a learned growth rate model, initialized via unbalanced optimal transport. This allows the framework to account for mass creation and destruction between time points, critical for modeling rapid embryonic development, cancer progression, or immune responses. Third, spatial context. A cell doesn't live in isolation. Its behavior is shaped by the neighborhood, its cell types, ligand-receptor interactions, and local gene expression patterns. MIOFlow 2.0 breaks new ground by constructing a joint latent space that fuses gene expression with spatial features. From each cell, it extracts four key modalities: the frequency of surrounding cell types, the strength of ligand-receptor signaling, local tissue density, and the average gene expression of neighbors. These are aggregated into a spatial feature vector, dimensionality reduced, and embedded alongside the transcriptional state. Now here's where the magic happens. All of this happens in a geometry-aware latent space, learned via a PHATE distance matching autoencoder. Unlike PCA or t-SNE, which distort the true biological relationships, PHATE preserves the intrinsic manifold structure of the data. This means distances in the latent space reflect actual developmental distances, not artifacts of high-dimensional noise. From there, MIOFlow 2.0 learns a continuous flow using neural differential equations, guided by optimal transport costs computed over geodesic distances on the learned manifold. The framework minimizes a composite loss, marginal matching, ensuring predicted cells align with observed distributions, transport energy, favoring efficient, low-cost paths, and manifold density regularization, preventing shortcuts through empty regions. And because it's trained on both gene and spatial features, MIOFlow 2.0 can answer questions no other method can. For example, can two cells with identical gene expression follow different fates? Yes, and MIOFlow 2.0 reveals why. It shows that their surrounding microenvironments differ, and those differences steer them down distinct paths. We validated MIOFlow 2.0 on synthetic datasets generated by SERGIO, a stochastic gene regulatory network simulator with known ground truth trajectories. On both a trifurcating differentiation process and an S-shaped trajectory involving cell cycle dynamics, MIOFlow 2.0 outperformed state-of-the-art methods like flow matching and Schrödinger bridges. The key difference: while others deviated into unpopulated regions of the latent space, MIOFlow 2.0 stayed tightly aligned with the data manifold because it learned the true geometry. An ablation study confirmed the necessity of each component. Removing the stochasticity term led to rigid, deterministic paths that failed to capture branching. Without the growth rate model, the framework couldn't handle population shifts. And without spatial conditioning, it missed critical environmental drivers. But the real test came in a biological case study: axolotl brain regeneration. Using a spatial transcriptomics dataset spanning 7 time points, we applied MIOFlow 2.0 to trace the trajectory from progenitor cells to mature neurons. The results were striking. We discovered that the abundance of medium spiny neurons in a cell's local neighborhood strongly influenced its regenerative fate. Cells surrounded by more MSNs were more likely to adopt a specific trajectory, suggesting that MSNs create a signaling niche that guides regeneration. This insight is only possible because MIOFlow 2.0 integrates spatial context into the dynamics. We also uncovered a key signaling pair, NCAN, SDC3, whose activity increased over time and correlated with neuron outgrowth. The model decoded this trend across trajectories, revealing how spatial signaling evolves during regeneration. This is not just about better predictions. It's about new biological discovery. MIOFlow 2 enables us to simulate what-if scenarios. What if we altered the local cell composition? What if we blocked a specific ligand-receptor interaction? The model can predict how such perturbations would reshape developmental outcomes. In summary, MIOFlow 2 is more than a trajectory inference tool. It's a unified framework for modeling cellular evolution, one that respects the data's geometry, accounts for biological realism, and integrates spatial context. It moves beyond interpolation to predictive, causal modeling of cellular dynamics. By bridging single-cell and spatial transcriptomics, MIOFlow 2 opens a new frontier in computational biology, understanding not just what cells are doing, but why, and how their environment shapes their destiny. This is the future of single-cell analysis, not just mapping cell types, but reconstructing the invisible choreography of life, one stochastic, spatially informed, dynamically evolving trajectory at a time.